Hello everyone, a very good morning to all of you. Today is the fifth and last day of our AICT sponsored AutoLFDP on cancer genomics in a healthcare system. Dear participants, I hope you have enjoyed all of their sessions, and honestly, we have tried our best to give you the best knowledge in particular in this field. We have contacted with all the top scientists, academicians, researchers, and experts in particular this field, and they have done their best to encourage you, motivate you, and to boost up your skills in cancer genomics. I hope this FDP will create a great impact on your scientific thought and your mind so that you can do something great in cancer genomics, also in cancer therapeutics. Today is the last day, and today is Saturday also. And as maximum of you, the participants are actually faculty members. So I believe that today you do not have any lectures, you are not busy with any academic curriculum, and today you will be able to enjoy last two sessions. And I'm very happy to tell you that today a very renowned two speakers will be giving their lectures. Dr. Akhilesh Mishra, he will be joining from USA, and also Professor Dr. Sanjay Kumar Pal, he will be joining from Nigeria. First lecture will be delivered by Dr. Akhilesh Mishra, and in India right now, this is morning at 10 a.m. But in USA, the time is very much different. In USA right now, it is probably the night, and I'm so happy that Dr. Mishra accepted our invitation, and he is happy to deliver the lecture despite his all busy schedule and in the night period. He is a very renowned genome researcher, and recently in COVID pandemic time he was in media because of his great discovery in COVID-19 diagnostic kit. These are the some news reports that you might have also seen in the COVID period, and I'm also happy to inform you that his diagnostic kit also right now in the market, which is also approved by the ICMR. Dr. Mishra, if I tell more, he received his PhD from IIT Delhi at SF Bio, and his expertise is actually ranging from various areas like computational genomics, also in analyzing the genome, and he is very good in understanding the genome. A researcher like Akhilesh Mishra is making all the things are much more easier. He has developed a lots of web servers like PSDDF, and now he is also working in cancer. His area where he is working in kidney cancer program as a postdoctorate research at UT Southwestern Medical Center, Dallas, Texas, in United States of America. He is also the qualified GATE, CSIR, NET, and also BINC GRF, that is exam. And he was also won the BINC award from DBT India. His research career was published with multiple research papers like in various journals like European Urology, Nucleic Acid Research, Biophysical Journal, Bioinformatics Method, and maximum of the things are associated with the genomics. Apart from this, Dr. Akhilesh Mishra was the managing director of Navadivya Nutrers Agro Private Limited, and we hope his effort is short. We hope short effort. Active experience will help all the participants to grow up in this field. But before going to his lecture, I also like to mention all of you that during the COVID pandemic time, he has done tremendous work to understand the COVID, that is SARS-CoV-2 genome, its all the various aspects, how it is associated with the disease and its origin also. They have published after the outbreak of COVID pandemic. He and lots of scientists in IIT Delhi, they have studied the genome very rightly, and they have published their paper in bioarchive. After that, it was a big sensation, and it was a big media attention for all over the not in India, all over the world. They got huge appreciation, and you can see in the news reports Dr. Anthony Fauci also forced to make his comment on particular their work. They have identified that they are about the motives, how they are associated with SARS-CoV-2. So with a small such introduction, let me hand over today's session with Dr. Akhilesh Mishra. Sir, I think has already joined, and uh, yeah, sir has already joined. So, sir, we are really thankful to you that, sir, you have joined us. And today, I think this is the night time in your USA. So, despite your busy schedule and compromising with your sleep, you are teaching us. We are really thankful to you, sir. Welcome to NIT, sir. Over to you, sir.
Good morning. Uh,
Good morning.
Morning, ma'am. How are you?
I am fine. How are you?
I'm fine. So
Okay.
it's night time, and like here I am in my lab.
Yes.
Uh, so it would be like first-hand experience. Um, Good morning to all participants. I hope all majority of you are like faculties. So it would be I'm a bit nervous to present my work in front of you, but I hope like it's science is like discussion. Science is about sharing our thoughts.
Yeah.
I will give my thoughts. You will give your thoughts. So we will discuss about like how we can improve the science. First, I would like to congrats Abhijit. Uh, I met him around 3 years before, 3, 4 years before. After that, he's doing so well. I'm very happy about him that he's working with such a renowned institute and organizing such a like in with Rupa Majumdar ma'am. Like he's doing so well. So I'm very happy about him, and I hope that my about a bit knowledge of my genome can give you a pinch of salt of experience.
Yes.
So without wasting any time, I would like to start my presentation. Uh, so can I share my just 1 minute? So, mm, until unless like I join, like there is a few seconds delay in joining because I'm not getting the link how to share my screen. By that time, if any one of you participant have some specific questions, some specific interest, please let me know so that I can include that in my today's presentation. Uh, so who's this? Uh, Dean Dayalan? Dean Dean. Yeah. So you have raised your hands. Like, do you have any question? So, Abhijit, I'm not able to share my screen.
Sir, in the top right, there will be
Yes.
one option. There will be one
There,
arrow
yeah, it
in
is
top right.
asking me
You
to
have to
browse.
click here, yeah, share content, and then there will be there will two things: screen and window. If
Yes,
you click on screen,
yes, that
if
I
you,
know. The problem is that it's just showing that it is browsing.
uh,
Not.
uh, s- so otherwise you can click on window. There might be lots of window.
Vijay, you can share the PPT also. You
Okay,
can
okay, okay.
take the PPT from Dr.
Okay.
That also we can do from our end.
Let me 1 minute. Have another screen. I can because like I have to say every time that please change the slide, go to that section. I just want to skip that thing. Just a minute. I'm connecting with another device.
Okay, okay.
Okay.
Mm. Is I audible?
Yes, sir. You're audible.
Yes,
Okay,
audible.
that's great.
Audible.
So just a minute. I have one presentation, so
Do you work here? It was wet.
yeah,
Do you
actually,
talk?
uh, just a minute. Uh,
Sir, if it is available on your drive, then I can also you can also send me the link so I can download and bring it. Uh, Dr. Mishra, if it is available in your drive, Google Drive or another drive, you can share the link so I can also present from here. You are mute, sir. Yeah, yes, sir. It is visible. Kindly unmute yourself, sir. I can see the screen.
is this now visible?
Yes, sir. Yes.
Yes, screen is visible now. Yeah.
I'm just shifting my system, so just pardon me for a few seconds.
Yeah, definitely. Nice. Actually, ma'am, right now the day on USA is 19th. And when I have spoken to sir that your lecture will be 20th, 20th has become
Yeah,
18th.
yes,
Just,
that's the confusion. So I thought my
yeah,
lecture is on
Tom-
20th. So
yeah,
wait for Saturday, and today is Friday here. So
yeah,
this
today is Friday news.
confusion, yeah, yeah. So nevertheless, we will start our presentation now.
Yes, sir.
So participants, good morning.
Yep.
Uh,
Sir, their audio is mute. They are writing in the chat box to wishing you a good morning. You can continue, sir.
And Dr.
thank-
Akhilesh, you can make it in the slide show mode also.
yes, yes.
Yeah.
Yeah.
So. So friends, so today I will discuss about my experience on physical, chemical, and structural characterization of genome and its application in cancer. So what I mean by physical, chemical, and structural characterization. So when I start in in my PhD work, I have done my PhD under Professor B.J. Ram from Supercomputing Facility for Bioinformatics and Computational Biology. In my PhD work, so when I started my PhD in genomics, the the major challenge that I face is that we do not know the language of DNA, how the DNA works, how the information is coded in the genome. So my supervisor, Professor Bijaram, asked me to work on this area, to work on that, to understand the language of DNA. So for this, I started working with the DNA as a sequence, then DNA energetics, and then the DNA structural parameters, and then its application. So what I mean by DNA sequence, so we will see that all the four dimensions of the DNA, that is the DNA sequence, DNA energetics, DNA structure, and DNA expression. So first, I will start with DNA sequence. The first uh, project that I started is OncoReglon. So what I mean by OncoReglon is, if you work on a cancer, if you work with the DNA, if you want to target DNA if you want to target a DNA, the major challenge is that let's say if you want to target a transcription factor binding site, majority of the transcription factor binding sites are of 9 to 10 or 9 to 12 nucleotide. Now, if you want to target that 9 to 12 9 to 12 nucleotide sequence in the human genome, it would be that particular transcription factor may be present from more than 10,000 positions in the whole human genome. So that is the major challenge. Like if you want to target any specific position, it should be targeted. It should be unique. Then only you can target it. So the first algorithm that I have written is OncoReglon. In this, what I have done is that this has 4 major modules: frequency counter, position finder, nucleotide sequence extender, and closer sequence finder. The uh, the frequency counter is let's say you have a 9 to 10 nucleotide amino acid sequence, the nucleotide sequence. You put that nucleotide motif, search it in the whole human genome. Let's say you get that motif in 10,000 position. Then the second step is position finder. In the position finder, you have to select a specific position. Okay, so I want to select the position which is present before P53 TP53 gene. I want to target this transcription factor binding site which is present before TP53 gene. So you should select that particular position. Then the next step is a nucleotide sequence extender. So in the nucleotide sequence extender, this is the software. Like in this module, what you can do is that you can just automatically it will fill out your particular position and it will extend the motif in that transcription factor binding site, that particular motif, in both the direction until unless its frequency will become 1, unique in the whole human genome. Now this particular motif is unique, which can be targeted by using either like by using as a by using siRNA or by CRISPR or by small molecule. So this software is to make us a particular position unique in the whole human genome. The fourth module is closer sequence finder. What that software means is that whenever you, let's say you want to do the binding, it might be possible that when you are targeting a sequence, it may show a off-site targeting. So what do we mean by off-site targeting is that it when we try to target a whole DNA sequence at particular position, what will happen that the last nucleotide, this side and that side, the terminal sequences have the lowest affinity. Because of that, what will happen that when you try to dock your drug molecule, it will show less specificity. So what if your unique sequence is of 12 nucleotide, and what if it show a lower affinity with last 2 nucleotide? In such a case, this particular module will give you the aspect of off target, like what are the off target site where the drug can bind, and you can do this study. Apart from this particular software also have a transcription factor binding site database for the cancer gene of about 938 genes, so that you can directly select a transcription factor, its transcription factor binding site. You can then put it in the frequency counter, then position finder, nucleotide sequence extender, and closer sequence finder. The result would be like if you put in the chromosome number 12, if you put this particular position of length 9, you can then this particular nucleotide sequence extender will extend it from 5' to 3' direction, 3' to 5' direction, and in both the direction. So in such a way that you will get, okay, like this particular motif ACGG, the 13 the final length 13 nucleotide, this particular motif is unique in the whole human genome. So if you make a drug target, if you make a siRNA or sgRNA against this particular motif, then it should be unique in the whole human genome and you will not get an off-target site. So this is my hypothetical aspect, that this is my study that I want to do. Before entering into the next step, because it's my bioinformatics, so I will also show you my software that I have developed. It's a free available software. If I show you, you can just go to Google, just type onco regulome. So as you can see, this is the software. You can work with GRCS 37 also and GRCS 38. Let's say you have selected GRCS 38. Here you can give your sequence, whatever sequence you have, A, T, G, C, T, G, A, T, G, C. Let's say you have this sequence, and then you can submit it for to find out the frequency of this motif in the whole human genome. Uh, my friends, if you have any doubt, please ask question. Please discuss with me. I would be more than happy to discuss your doubts, and will try to. So this is the software you can use after my lecture, and if you have any doubt, you can email me. So this is the software that I have developed. This is the cancer gene database that I have developed. So and it is freely available software that is developed by SCO Bio by me. After that, uh, the in the DNA as a sequence only, the next software that I have developed is pathogen specific DNA drug finder. Pathogen specific DNA drug finder. What does it mean by pathogen specific DNA drug finder? So do anyone have an idea that a pathogenic strain of a genome, let's say if you talk about the E. coli strain, a pathogenic strain and a non-pathogenic strain, what is the difference in the genome? Like what is the percentage difference? Anyone have any doubt and any idea?
So there should be some gene which will be after translation, which will be creating such protein which might be harmful for us, but not for themselves.
Okay, yeah. So having a novel gene is very rare. Like if you talk about the two transcription factors, and if you say that in this particular, yes, there is a horizontal gene transfer, there is a uh, the resistance, acquired resistance from the environment. Those all things are there, but if you see by difference, the 99.5% of the genome of a pathogenic strain and a non-pathogenic strain remains same. The only difference is not more than 0.5%. So the question arises that what is that 0.1 or 0.2%? In general, it's 0.1 to 0.2%. What is that 0.1% of the genome which is making that particular strain pathogenic? This question arises in my mind, and I try to find out the answer that how can I find out that 1.1% genome which is different from the non-pathogenic strain and provide the pathogenicity, provide it resistance from the antibiotics, provide it lethality, like what not, like that 1 point, what is that 0.1% genome which is causing the disease? For that, I developed a very simple software. In that, what I have done is that I have compared the pathogenic strain, the pathogenic genome with the related 10 non-pathogenic genome. Then, after comparing the pathogenic genome with 10 non-pathogenic genome, I tried to find out what are those motifs. After finding out those motifs which is present in the pathogenic strains, like that 0.1%, and that is absent in the non-pathogenic strain, just to confirm that this 0.1%, like this particular motif, is responsible for the pathogenicity. So I I have done the comparison. I have done again an analysis to compare this motif with these other pathogenic strain. So first I've compared my pathogenic strain, the pathogenic strain of my interest, with 10 non-pathogenic strain, and then this pathogenic strain with other pathogenic strains, other related pathogenic strain, to find out that is this motif is present in the other pathogenic strains or not. After that, because that particular bacteria, after finding out that particular motif which is absent in all the non-pathogenic strain and present in pathogenic strain, and because that pathogen is also present in the human body, so we have to extend that particular sequence in both the direction until unless its frequency will become 0 in the human genome. so that if you make a drug against that, it should not target the human DNA, because the pathogenic DNA, pathogenic genome is also made up of ATGC, and the human genome is also made up of ATGC. So after making its frequency 0 in the human genome, now this particular motif, we can target it by several ways. First, if the motif is a part of a series, if the motif, so if the motif is a part of a series, then we can, if it is a series, then it will translate into a protein, and we can target the protein structure or transcriptome by using siRNA, miRNA, or shRNA, and most preferably by using the small molecule protein docking. What if it is not a part of a series? If it is a part of a regulatory genome, regulatory part of the genome. In such a situation, what we can do is that we can target the DNA motif by using antisense RNA, CRISPR technique, PNAs like protein nucleic acid sequences. So there are several techniques that we can target that particular motif which is responsible for the pathogenicity. Or we can, so this particular pipeline is developed by in SCA Bio only, like by me and by my colleagues. In this, we can so this is a software called RASDD. By using this particular software called RASDD, we can screen out the the zinc database molecule which can bind to that particular structure, which can bind to that particular DNA sequence. And then after binding, we have two further softwares called minor group binders and DNA intercalators. So what is called minor group binders? So if you want to target a DNA sequence with a drug molecule, there is two strategy. So one is called minor group binder, and another one is DNA intercalator. What is minor group binder? So as you know, the DNA structure has a minor group and a major group. Now this, so generally, majority of the DNA targeting drug available in the market are the minor group binders. Now, this particular minor group binder binds to the DNA molecule, and either it breaks it, or it won't allow the RNA polymerase to move on and stop the. So generally, these software are for the cancer therapy, which actually binds to the DNA and stop the replication process. The other way of targeting the DNA is DNA intercalator. What do we mean by DNA intercalator? So as you know that in the DNA, we have two interbase pairs, like this is AT, this is GC, and you have a space between this base pair and this base pair. So between two base pair, the space is called if your drug can enter inside this, like as you can see here, if your drug can bind here enter inside here, this is called intercalation, and this is called intercalator. So we have the software for minor group binder also, and the DNA intercalator both. After that, after binding with the DNA minor group binder and with the intercalator, we want to calculate we would like to calculate the free energy, that what is the affinity between the DNA and the drug molecule. For that, the software is called Predicta. This is also developed in India by SCA Bio, by my previous lab, and by using this particular software, you can find out the lead molecule which can bind to specific DNA sequence. By using the same strategy, I work with the mycobacterium. First, I have made a mycobacterium species, non-pathogenic mycobacterium species list. So these are the 10 mycobacterium species which are non-pathogenic. And then I have compared the Mycobacterium tuberculosis S37RV with all these 10 non-pathogenic strains of the Mycobacterium. And then these are the other pathogenic Mycobacterium species. So we have 10 non-pathogenic strains and 10 pathogenic strains. And we have compared first this Mycobacterium tuberculosis S37RV with the non-pathogenic strain, and then with these pathogenic strains. What we find out is that we are able to find out a motif, that is TADTATAA, which is present at this particular position, which is absent in all the pathogenic genomes and present only in absent in all the non-pathogenic mycobacterium genome, and present in majority of the like 8 out of 10 pathogenic strains. So we find out that, okay, this particular motif, just a minute, there's a light issue, just 1 minute. Hello? Hello? Yes. So so now this particular strain, pathogenic, this particular pathogenic strain. So we have find out this particular motif, which is yeah, which is absent in all the non-pathogenic genome and present in only in the pathogenic genomes. So now we are in a procedure, like at that time, now our lab is also collaborating with the wet lab person, and we have find out that this particular motif is a part of a PPE 54. This protein is not well studied protein, but we know that the PPE family is very much responsible for the pathogenicity, to provide the pathogenicity to the Mycobacterium genome. After working with oncoroglan, PSDDF, and other aspect of the DNA as a sequence, the question that arises inside me is that, is the sequence is sufficient? Is that when I'm talking about to bind a DNA sequence, to target a DNA sequence, how we can target a DNA sequence if you do not have structure? How those molecules which are present in the market binds to the targets the DNA molecule if the DNA is only a sequence, if we do not have any structure? Further, the question what arises in my mind is that the DNA, so if you like how a protein interacts with the DNA, is it only the sequence? So this question arises in my mind. Then I started working with the promoters in the prokaryotes. And what I have done is very simple experiment. What I have done that I have taken 10 promoters from 10 different genomes. And I make a multiple sequence alignment of the promoter region. What I found is that there is no consensus. There is no consensus out of these in the 12 promoter region from 12 different organisms. There is no consensus. But if you use a RNA polymerase of one organism, and if you use that RNA polymerase to the other organism, it works well. Then the question arises, if the sequence is not same, what is that thing the RNA polymerase are recognizing? What is that thing that RNA that the protein are able to interact with the protein, interact with the DNA? This question pushed me to work on the other aspects of the DNA. But as you know that in like, so just for like a discussion, I want to say, like, if there is no consensus in the 12 promoter region from 12 different organisms, but if you discuss that this Escherichia coli, in the Escherichia coli you can see that you have a consensus at -11 region, TATA80, and this that the Pribno box and Tata box. If you talk about the Thermococcus, there is a consensus at -37 region of TTTAAA. If you talk about a Mycobacterium tuberculosis, there is no consensus. Even if you take a Escherichia coli, and if you just for your experiment, if you try to do a multiple sequence alignment between all the promoters of the Escherichia coli, you will surprise to see even 40% of the sequences do not have Tata box. So how the RNA polymerase are interacting with DNA? If sequence is not if sequence is not same, if the sequence is not the only thing, then I started working with DNA energy parameters. In the energy parameter, the first three parameters that I have discussed is hydrogen bond energy, stacking energy, and solvation energy. And I used a very simple formula for calculation of hydrogen bond energy, that is total hydrogen bond energy divided by number of dinucleotide. Like, just it should not be biased because of length. So that's why divided by number of dinucleotide. So total, so it's a dinucleotide thing, addition of hydrogen bond energy and divided by number of dinucleotide. So this is the very simple software formula that we have used. And similarly for the solvation energy and stacking energy. And what we have used is that we have used about 7.6 million genes from the NCBI. That is the database, that is the whole database available at that time. We have considered 7.3 million mRNA, 2 lakh, around 2 lakh 0.25 million tRNA, 5,000 miRNA, 3,000 snRNA, and like around 13,000 16 sRNA, 23 sRNA, and 5 sRNA. And you will be surprised to see that we are able to distinguish a different functional unit of the genome, that is the mRNA, tRNA, siRNA, snRNA, and the different subunits of the rRNA, just on the basis of the energy parameter. What do I mean by that? I mean is that the DNA, and this is not at the RNA level, this is at the DNA level. So what I mean is that at the DNA level, mRNA knows that I am mRNA. tRNA knows that I am a tRNA. So their energy profiles are different. The tRNA have a different energy profile in comparison to mRNA, siRNA, miRNA, and other subunit of the genome. And we can identify the energetics by using just three parameters, that is hydrogen bonding energy, solvation energy, and stacking energy. So you all have an idea about what is hydrogen bonding energy. What is solvation energy? Anyone wants to interact with me? Like anyone want to discuss? What is solvation energy and what is stacking energy? Uh, Anyone? Um, so, Abhijit.
Yes, sir.
Yeah, I just want to be sure that that the participants are able to like hear my voice and like.
Yes, sir. They can hear you. Actually, they cannot unmute. When the setting was done, they can only type in the chat box.
Okay, okay, I
Yes.
understand. I understand. Okay,
Yeah, Dr. Dharmendra Kumar is writing. Yes, sir. He's understanding
okay.
in the chat
Thank you.
box. Yes, ma'am.
Thank you. Thank you. Yes. So the solvation energy is that when is something about the hydration energy. What if I put a RNA molecule inside a water inside a water system, inside a solvent? What will happen? How the solvation energy will work? So if a particular structure of a RNA is linear, its solvation energy is always be higher in comparison to those which are which have a combined structure, which have a 3D structure. For example, tRNA. So tRNA have a clover leaf model, but mRNA are almost linear, almost circular. So that's how the solvation energy works. What is stacking energy? So stacking energy is if you stack a coin of one coin above that. So this is called stacking energy. So by using these three energy parameters, we are able to distinguish these different functional units of the genome. After that, so at the same time, like when I'm working with the DNA sequence, when I'm working with the DNA energetics, at the same time, I'm also working with my dream project that is called ChemGenome. So in ChemGenome, we are trying to annotate a prokaryotic genome based on their physicochemical properties. So earlier, we were only working with the DNA as a sequence. After incorporating DNA energetics, we are able to increase the accuracy of genome annotation software about 10%. Like earlier, we were at 60%. After adding the the DNA's energy parameters, we are now at 75%. But still 25% are remaining. We are not able to say, we are not able to identify what is that 25% is. So I started working with structure of the DNA. You all guys are uh, very familiar with the structure of a protein: alpha sheet, alpha helix, beta sheet, loop, and all other. But what do we mean by structure of DNA? When we say about the structure of DNA, what we think is that having a 10 angstrom helix diameter and like a major groove, minor groove, 3.4 angstrom of axial rise, this is the DNA structure. Or what we think is the conformation of DNA, like A DNA, B DNA, Z DNA. We think that this is the structure of the DNA. But my friends, this is not all. The DNA structure is much more than that. We have 9 backbone angles, that is alpha, beta, gamma, delta, epsilon, chi, phase and amplitude. We have 8 interbase pair parameters, 6 intra-base pair parameters, 5 base pair axis parameters, and 3 energy parameters. That is 31 parameters we have. So what do we mean by interbase pair parameters? So interbase pair parameter means is that when you have this AT and this GC, like so if whatever is happening between these two, so all of you are able to see me. Is my video uh, is visible?
Yes, sir.
Abhijit?
Visible.
Okay.
Yes, sir.
Yeah, yeah. So if you say this thing, this is AT, this is GC. If you say this, and if so, this is called as shift, or this is called tilt. If I reduce the angle, this is called tilt. Twist, inclination, tip, rise, slide, shift. These are the interbase pair parameter. Then if this is A and this is T, this is called intra-base pair parameters. You understand, guys? This is inter, this is intra. Backbone angle is like alpha, beta, gamma, delta, epsilon, chi, phase and amplitude. These are the 9 backbone angles. And then we have a base pair axis. So if we have this backbone, we're having this sugar ring, and then there is something here which is attached with the backbone. So what is this? Like this angle, this is called base pair axis. So we have around 28 structural parameters. So if you can see that this is the alpha beta, like if I show you this particular angle, let me show you with through pointer. This is alpha, this is beta, this is gamma, this is delta, this is epsilon, this is chi, this is phase, and this is amplitude. So this is how torsion angle of nucleic acid structures are. To study on this, as I said, that I worked with the 12 different organisms. What I have done is that I have considered around 12 organisms, including 3 archaebacteria and 9 eubacteria, because we consider that the archaebacteria are a bit different from the eubacteria. Now we have made this study with the largest ever known data set of 16,000 promoter sequences to find out the structural aspect of the DNA. What we have done is that we have 9 backbone angle, 8 interbase pair parameter, 6 intrabase pair parameter, 5 base pair axis parameter, and 3 energy parameter. And my friends, I'm surprised. At that time, like my eyes are like, I'm very new in research, and my eyes are full of tears to see this. I have seen that DNA wants to talk. This particular we are seeing at 0, this is the TSS. There is no consensus at the sequence level, but when we are working with the structure and the energy aspect of the DNA, there is a clear signal that this is the TSS. So there is a conservation at the structural level, but not at the sequence level. So whenever we talk about a DNA sequence, without structure, it is incomplete. Then I worked independent with all the 31 parameters. Like if you can see the 9 backbone angle alpha. So this red line represents the CDS sequence and the green line represents the promoter sequence. So you can see that in all the 28 parameters till here and these three energy parameters with the with in 16,519 nucleotides, we can see that the signals are so clear that here is the TSS. Here is the boundary region. Like if you see alpha, beta, gamma, delta, epsilon, zeta, chi, phase, amplitude, shift, slide, rise, tilt, roll, twist, H rise, H twist, shear, stretch, stagger, buckle, propel, opening, X displacement, Y displacement, inclination, tip, X spin, and hydrogen bond energy, stacking energy, and solvation energy. That a question arises that is that all the parameters are same? Is the signals are same in all the prokaryotic organisms? And when I worked at an organism level with 12 organisms, 3 from Archaebacteria and 9 from Eubacteria, what I found is that all these parameters are able to find out the signal, but the signatures are different. What that means is that the language of DNA, that is ATGC, is same. The structure is same from virus to human. From virus to human, it's ATGC. It's the same structure. It's all same, but the story are different. The signature is different. Language is same. They all are written in one language, that is the language of God, that is DNA. And this DNA language, the story is different for each organism. That's why when you work with any other genome annotation software, which is all majority of the genome annotation software are made by machine learning or by hidden Markov model by training and testing algorithms. In that particular situation, what will happen is that because you are training your model in one set and you are testing it in another set, in such a situation, what will happen, because the signatures are different, you will see that as you try your model in a new organism, the accuracy dropped very drastically. That's why there is a need to work on the physicochemical parameters to identify the features, to understand that how the story, that how what is the language of DNA. You can see that for just like you can see for Mycobacterium tuberculosis, it's so precise, like just 10 to 15 base pair before, 10 base pair after, and the signal is over. But if you see here, like in Chlamydia pneumoniae, the signatures start from minus 200 base pair and ends still 100 base pair. Like this, that's how the difference is. These are the TSS profile of 12 different organisms. If you then one question also generally come in our mind, and what we think is that if the protein which actually guide DNA, it's the protein who recognizes the TSS region. My friends, it's a very long time, it's a big debate that who has the property, like is it a protein who identify the DNA or it's the DNA who identify a protein. So in that, what I found out is that the DNA has its own property. The DNA has the property to say that this is the TSS, and how it works is that if you take a, if you that this is how it works is, just try to understand. Let's say this is one, let's say this is base pair axis. Now, in the base pair axis, the DNA structure, what will happen? Uh, let's say we have this thing. Now, if I twist, high twist at this particular region, and if I have a high twist at this particular region, what will happen? If I twist it from this direction and this direction, what will happen?
Obviously, sir, some energy changes and structural
Yes.
changes.
So what will happen is that if you have high twist low twist and high twist. If we have such type of combination at the TSS region, what will happen is that it will actually, because of this high twist, the middle low twist region will come out. And this is how the thing is working. Like there is a high twist, low twist, and high twist. When because of this two high twist region, this low twist region come out, provide a position where an aromatic shaft of the sigma protein of the RNA polymerase can go inside it and initiate the transcription. And that's how the transcription start from -11 region in E. coli. Then, after having this knowledge, my next, because I'm a bioinformatician and my role and it's my duty to develop softwares for you guys. So I started working with to develop a software called SCPROM. So with the backbone parameter, with the inter-base pair parameter, intra-energy, and base pair axis parameters, we have developed a software called SCPROM. That is SCPROM, a prokaryotic promoter prediction software. And I can tell you, my friends, that the accuracy what we have, the sensitivity, the F value and precision, which represent the sensitivity and specificity, we have the SCPROM have the maximum sensitivity available to any other software in the data in the literature, and has the highest specificity in highest sensitivity and specificity in comparison to any other software which present in till now. We have shown by using the structural and energy parameters, we are able to find out the TSS region much more precisely in comparison to any other technique. Now, like we have the algorithm is bit like it's regression based and PCA based. Like with the regression algorithm and the probability based model, we have tested this thing, and like we have made the algorithm. The software is freely available in the name of SCPROM. You can download it and use it. If you have any doubt, please email me. I will love to help you out. You can see that in the now the performance of SCPROM in promoter identification of 12 different organisms. You can see that we have 83% sensitivity and 80% specificity. My guys, even after having the maximum sensitivities and specificity present in all other, in comparison to all other softwares in the literature, still we are at 83%. Still, we do not know what is that 20%. What is that 70 to 20% thing? For that, we are working with we are working with an international collaboration. Now I am in US. My junior Dinesh is now working in this project, and he is collaborating with Modesto Orozco, well-renowned bioinformatician, like very big professor from Spain, IRB Barcelona, and Richard Lavery from France, from Beveridge from Washington. So like now we are collaborating with these persons to work with because this particular model is on dinucleotide now, and but on the DNA structure level, crystal structure. Now my friend, like my junior, I'm also in that project. I'm also helping him to resolve it. So now he is working with tri and tetranucleotide to increase the accuracy. After that, after working on the prokaryote, as I said, the language of the DNA is same from virus to eukaryotes. The question from virus to human. The question arises that can we apply this knowledge to humans also? So in humans, the question is the biggest mystery is the intron exon boundaries. If someone asks you that that a wheat genome is much longer than human, but still the complexity, but still we are more advanced, we are more superior. How? The question arises, the answer is that what we call about C-value paradox, that as the genome size increases, complexity do not increase. This disbalance is called C-value paradox. The question arises that this C-value paradox thing, like how we can define, how we can resolve this thing. So the thing is that those organisms which have higher level of complexity are more complex in spite of whatever their genome length is. First. Second, so what do we mean by complexity? So complexity means if you take a human gene, or a human gene can give rise to more than 10 different isoforms by the name of different transcripts. The complexity of the human genome is hidden behind this intron-exon boundaries. This is the biggest mystery of the human genome, to identify the intron-exon boundary. When we have, and there is no software till now available to 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 identify the intron-exon boundaries. When we try our energy and structural aspect in this particular software, like where we have this energy parameters, like we have the 28 structural and 3 energy parameters at the intron start site and intron end site, in comparison to the serious reason, we are able to find out that this is the reason, like the signals are very clear. You can see before 0 there is a signal, after 0 there is a signal, which shows that there is a very clear signal at the TSS region, which actually define the intron-exon boundary in the human genome. And same as in other eukaryotes also. After that, my friends, my dream project, that is ChemGenome, like this all sequence, energy, structure, these all things we are incorporating to make a genome annotation software which can be applicable to prokaryotes also and eukaryote also. And that could also be, and that is ab initio based, which can be used in any novel genome. So in that case, like by using the energy, homology, and DNA structure, we have developed ChemGenome 2.1, and now we are moving toward ChemGenome H to develop a genome annotation software. After that, I finished my PhD, and it's the beginning of the COVID era. So I decided not to move outside, and I keep working in the IIT Delhi as a research associate in under Professor Vivekanand Perumal in Kusma School of Biological Sciences. In that, I'm working with my major role is to work with the sequencing, to sequence uh, uh, the the to sequence the antimicrobial resistance. This is a project on rapid diagnostic solution to combat antimicrobial resistance. So I worked on it to identify the antimicrobial resistance. So during that, we worked on COVID-19. During COVID as in a research associate role, I contributed to the develop of India's cheapest COVID-19 diagnostic kit. It's 300 rupees kit, and got approval from ICMR, inaugurated by Honorable Education Minister Ramesh Pokhriyal. 10 big companies acquired the technology for manufacturing and marketing. And I can say with a proud that my group have never thought about money. We have sold it in as low price as it is possible. Like we have sold the license for 10 lakh rupees, one license, not more than that, just so that it should be the cost would be low, and it should reach to every people of India. The kit is based on pro-free real-time PCR. At present, like we are working with the mutational landscape of SARS-CoV-2 also. This is the kit that we have developed. At that time, its cost is 399 rupees. So, my friends, this is about the journey of understanding the DNA, the sequence, energy, and structure. Now, because this workshop is on cancer, I should talk about cancer. So now, my friends, after working there, I moved to US because of multiple reasons, and I joined kidney cancer program. It's the one of the largest lab in the world which works on kidney cancer. Uh, he works on kidney cancer and other patient data. So in that, the first project that I worked on is active surveillance. So what is active surveillance? I will like correlate what is like But first we would understand what active surveillance means. So the aim is to characterize the time to initiation of the systemic therapy in patient with metastatic renal cell carcinoma under active surveillance. What it means is, for let's example, if someone got diagnosed by cancer, in such a case, what will happen? If someone is diagnosed by the cancer, it should not be start treated with the radiotherapy and chemotherapy and all those from the next day. No, doctor do a active surveillance. So surveillance means it is on, like it's on scrutiny. Like it's got tested that is cancer is increasing its size or not, is the cancer is benign or malignant, when it is moving to the metastatic, when it need a radiotherapy, when it needs a chemotherapy. So that particular time when the patient is not medicated, but it is just on surveillance, it is just on scrutiny, this particular period is called active surveillance. And this period is very important for a patient life, because once the treatment is started, once the systemic therapy is started, the what will happen in cancer, like it will lose all its hair. It's a very painful thing. So my first project here is to understand the active surveillance. Earlier it was understand that, like, so how to identify that when this patient will what would be the active surveillance period for this particular patient. So to identify that signature motive, we worked on that and we find out that it's 14.5 months were considered as the average time for active surveillance in renal cell carcinoma. So if someone has renal cell carcinoma, with some day, like the average period is 14.5 months, but in some cases it is 3 months, some cases it is 64 months. It's varying a lot. How to identify that? So in that, first we worked with the 52 patients, and in that, like, we sequenced the whole exome sequencing of the patient data, that the sample directly taken from the patient tumor. And when we have performed the whole exome sequencing study, what we have find out that TP53 gene and SMARCA4, TP53 and SMARCA4, these two are these two proteins. If any of the patient have mutation at TP53 and SMARCA4, then then the time of active surveillance will reduce drastically. See, if you can see that till here, like, when there is a mutation in the TP53 and SMARCA4, the active surveillance period is even less than 6 months. Then this is called as Kaplan-Miller plot, which present the this is Kaplan-Miller, which present the survival probability. We have seen that if the TP53 or SMARCA4 is mutated, then the survival probability also reduces. So what we have diagnosed is that if any of the patient came to our clinic, what we can do, we can just sequence the genome. We can see that there is a mutation in TP53 and SMARCA4. If there is a mutation in TP53 and SMARCA4, the surveillance period should be less than 6 months. And that's how we define the active surveillance period and the systemic therapy time. After that, we also work with the RNA-seq data. Like, after having the whole exome sequencing data, we worked with the RNA-seq data in the whole human in the patient genome. And what we have find out, that we have find out 17 such genes which can be used as a biomarker of early progression. So there is two things. One is called early progression and one is called late progression. So if the if the therapy starts before 1 year, we call it early progression. If the therapy starts after 24 months, we called it as late, longer or late, shorter or longer time of active surveillance. We find out these 17 genes which could be used as a biomarker to in the blood only, or in the RNA-seq, the tissue sample only, that if if the expression of these genes are high, for example in LEX1 or TSPR or QPRT, then it means that it has an early progression, and we should reduce the time of active surveillance, and that patient will very shortly should be put on the systemic therapy. After that, I worked the other project that I'm working in my lab is the TRCC project, that is translocational RCC. What is translocational RCC? So translocational RCC. So Abhijit, how much time I have?
Sir, actually, you almost completed your time.
Okay, I'm enjoying. So
Ah,
yeah,
yes,
I
sir.
will take just 5 minutes more. So what is translocational RCC? So translocational RCC is that, like, this is the patient sample and all those things. I will just explain what is RCC. So the translocational RCC is what will happen is that it's a renal cell carcinoma which happened because of fusion protein. So what will happen? There is a one protein called TFE3 protein, TFE3 and TFEB. When this TFE3, TFEB protein got fused to the different fusion partners, this particular fusion lead to a cancer. Just one fusion lead to a cancer, and this particular and it cost about 50% of the pediatric cases in cancer. 50% renal cell carcinoma cases in pediatric patients, in children's, in renal cell carcinoma. So we are working on that, and like we have different models called PAC8 model, SGLT2 model. These are the mouse model where we are able to reproduce the cancer in the mouse. So that's a mouse model of PAC8, that is the embryonic model, and SGLT2, that is the adult model. And then we have around more than 50 patients of patient samples, like primary patient data, to find out that what is the mechanism behind translocational RCC. We are very close to that, and I hope by within 2 or 3 months, like 2 months, we are able to propose a medicine against TRCC. In my lab, more than 5 clinical trials are happening. Like we are playing very front forward to have we are like to diagnose the disease, to give a new drug molecules and all those. So like I hope like by within 3 months we are able to diagnose the TRCC, the mechanism of TRCC, and the drug against that. Thank you, my friends. These are few relevant publications that I have published in last 5 years, and my goal that one day everyone can read the bacterial genome or a human genome like a storybook. Thank you.
Thank you, sir. It was a great lecture. Honestly, I'm telling you, sir, as of now, our speakers, they are telling only about the sequence. And like Dr. Ritu Gupta and other our very renowned scientists, they have mentioned that that cancer in when one patient it is expressing in one way, but in another person it is expressing different way. But today from your lecture we are able to clear that not only we have to focus on sequence but only also in the structure.
That's true.
So we have we have to work a lot, means.
Uh, so Dinesh, is
Yeah.
there a possibility that you unmute the participants?
So actually, I actually said it will be able to done by admin, and we have received
Sorry.
one question.
Yeah.
We have received one
Yes. Yes.
question.
Yes.
Yeah, Dr. Sumaya Das ma'am is asking, how can differentiate in cancer cell metabolism to be exploited? Are mouse models still relevant to study human cancer?
Yes.
Huh?
So there is um, okay. Uh, Repeat the
Actually, there are
question one. There's
yeah, there are
repeat
two
one.
questions. Yeah, there are two questions. First question was, how can differentiate? How can differences in the cancer cell metabolism to be exploited?
Cancer cell metabolism to be exploited.
Yes, sir. Is there any bioinformatics solution as of now today? Or
Yes, like we do a metabolomic study. So what we will do is that different bacteria, different cancer have a different approach. As we know that the cancer have 8 different hallmark signature. So what we will do is that we will take a cell, the cancerous cell, and we provide uh, uh, radioactive uh, biomolecules to feed them, because as we know that the cancer have a very high metabolomic rate. What it will do that it will consume all the biomolecules in that cell, in the cancer cell, and it break the biomolecule and reuse it for its own use. Now when it break the biomolecule, and when it use it for different structure, for example, we have provided a nucleic acid, like you have provided the purine, like the purine pyrimidine bases. When it when it try to reuse it, the software reuse that biomolecules, then we will try to identify, okay, it is using the whatever the food, whatever the resources it is getting in this direction, in this particular pathway it is putting its energy. So that's how, by using this particular approach in here, we are identify that how, like by using the radioactive biomolecules, we identified that which pathway is most actively upregulated. Yeah.
Yes. And another question, are mouse models still relevant to study human cancer?
Yes, very relevant. The reason behind that is because we cannot, like the patient samples are very limited. We cannot get biopsy very frequently. So what we will do is that this particular procedure called PDX model. So in the PDX model, what will happen is that we take a mouse model, we take a patient biopsy, we take the patient primary tumor or the metastatic tumor, the primary tumor from the patient, and we put it into the mouse kidney.
That's it.
And then once this particular primary tumor is engrafted into the mouse tumor, and now it is within the body, we can multiply it. Now we can do the drug testing, we can do multiple type of analysis over that, like the drug analysis. And now the sample is with us, and now because that cancer is also now growing, this that particular cancer is also growing. So now we have a plenty of sample for RNA-seq analysis, whole exome sequencing analysis, and like multiple analysis. But the patient samples are very precious. We have very less amount. So the PDX, so this is the one region, the PDX model. The second model is, as I'm talking about you, with TRCC. So in translocational RCC, we have GEM models. So what is a genetically modified model? So what we will do, as we know that this particular cancer caused because of fusion between a TFE3 and with a different protein molecule. So what we will do, that this fusion complex, this fusion protein, we insert inside a genetically modified mouse, and then that mouse breed. Now because this mouse have this fusion protein, it also gain cancer. And now
Right.
this cancer, because of this particular fusion protein, it gained cancer. And now this model is good for all type of understanding the biological phenomena, for understanding the drug study, and all other studies. Yes, so the mouse models are very important. Yeah.
That's it.
Thank
Assalamualaikum
you,
everyone.
Dr. Mishra.
Team
Thank you. Thanks a lot
Yeah.
for sharing your very elaborative and informative knowledge on physicochemical and structural characterization of genomes and its application in cancer. We know it's not possible to share your entire experience and research work in a span of only 1 hour. But of course, we have a plan to invite you repeatedly in our future programs also. And the participants, I think they are very happy to get you within them.
Thank you. Thank
Thank you
you.
so much. Thanks a
Thank
lot.
thank you. If you, to all my participants, to all my friends, if you have any doubt on cancer, genomics, or in structural and energy aspect of the DNA, please email me. You can get my email ID from Abhijit and Rupa madam.
Yeah, I'll share.
And
Yeah,
coming
of
to,
course.
thank you, Rupa madam. I have taken I
Thanks,
think
thanks a
10
lot,
minutes
sir.
more than what is allocated to me. But like I'm very,
Thank you.
yeah, I
It's
enjoyed
our
that.
pleasure to have you with us today.
Yeah, I enjoyed my presentation.
Of course, sir. Thank
thank,
you. Thanks a lot.
thank you, Vijay. Thank
Actually,
you.
sir, actually, sir, we have started slightly late, and you are actually on your time. So
It's okay.
Yeah, thank you, sir.
Thank
So,
you.
yeah. So now I like to request my colleague Deepika Pathak ma'am. I want to hand over this session to ma'am.